Hi, and welcome to this mind map. We're going to be reviewing everything in OCR A-Level Biology Module 5.7, and that's on respiration. So in this mind map, we're going to try and get everything on one big A3 page. So if you can, you could take a screen grab of this. I'll just disappear for a second and print it in A3. Or if that's not possible, you might want to take a screen grab of this. I'll disappear for a second. Print it out, cut the pictures out, and kind of create your own A3 mind map using those images, okay? So in this mind map, we're going to cover aerobic respiration, anaerobic respiration, the processes of glycolysis, the link reaction Krebs cycle, oxidative phosphorylation, and we'll also talk a bit about experiments that you can do involving respiration, involving yeast, and the use of a respirometer. And finally, we're going to be looking at respiratory substrates, and a little bit on ATP as well. Okay, so let's get into it. Okay, so here's our mind map, and let's zoom in, and we're going to start kind of in the center. We're going to start with this molecule, ATP. So ATP is kind of the Y of respiration. ATP is the molecule that is produced by respiration. So at GCSE, you may have said respiration releases energy. That's true. It releases energy in the form of a molecule called ATP, or adenosine triphosphate. So let's label our molecule of adenosine triphosphate. So ATP is a molecule that looks like this. It has three phosphate groups here. So these are three phosphates, all these here. And then this here is called adenosine. This bit here is called adenosine. And adenosine itself is comprised of an adenine base, which is that, and a ribose sugar, which is a pentose sugar. So it's a modified DNA base, basically. It's a nucleotide triphosphate. Now, the great thing about ATP is you can cleave phosphates off the end of it. So if you cut off one phosphate, you make ADP. If you cut off two phosphates, you make AMP. So let's show that here. So if I take ATP and cut it to make ADP, an inorganic phosphate that's floating off, I get some energy. And the amount of energy I get is 30.5 kilojoules per mole. And if I do the same thing again, and I take ADP and I cut off another phosphate, then I will produce adenosine monophosphate plus another PI. And I would release again that same value, 30.5 kilojoules per mole. And if I really wanted to, I could just cut off another phosphate and go to adenosine itself without any phosphate. And that doesn't give me quite as much energy. It gives me a little bit less. It gives me only about 13.8 I've got here. I don't think you need to know these values, but the reason for these high values of energy at the start is because all these phosphates are negatively charged. You can see here, I'm going to highlight in yellow, these negative charges. So they kind of are wanting to repel. So if you make a little cut, right there at that oxygen, basically what happens is the phosphate kind of flies away, releases energy. So this is why ATP is so useful. It is a useful molecule for kind of storing energy. So we would say, I'll switch colors, that ATP is the universal energy currency. Universal energy currency of living cells. What do I mean by that? Well, I mean, cash is almost out of date, isn't it? Most people use contactless these days, but back in the day when people used cash, if you had like a £20 note, that might have been annoying because it was a large chunk of money and it might have been easier to have pound coins. Imagine it like this, actually. Imagine you go into a shop and the shop gives no change at all. You're not going to use a £20 note to buy a can of Coke. you want to have something smaller. So you would try to break that £20 note up into £21 coins, and then if you went into a shop which didn't give change, and you paid for a can of Coke with £1, well, you'd maybe lose a little bit depending on the price of the can of Coke, but it would be a lot better than paying for it with 20 quid. So this is the idea of ATP. Glucose is like £20, and ATP is like those pound coins. So respiration is really breaking the big chunk of energy, the £20 note, into lots of small useful pieces of energy that can be used in the cell. So we call that, those sort of useful pieces of energy, that's ATP. So that's why we say it's an energy currency. So let's make a couple of notes on that. So it releases energy in manageable amounts. Releases energy in small, manageable amounts. It's stable enough in the cytosol, but releases PI, which is that phosphate, the inorganic phosphate. The I stands for inorganic, but releases PI and energy in a single reaction. So enzymes can come along and just cut off a phosphate, a little burst of energy, which can be used. And therefore you kind of release less energy. You waste less energy when you're using ATP. Now, what about making ATP? Well, there's two ways. Well, basically, you take ADP plus PI and you convert it back into ATP. And there's two ways of doing this. Number one is called substrate level phosphorylation, which is a simple chemical reaction. And the other thing is called oxidative phosphorylation that uses this molecule down here. So let's do oxidative phosphorylation first. Oxidative phosphorylation. And actually, oxidative phosphorylation is both this diagram over here and this diagram over here. So that is quite a complex thing. We'll get to that later. So that's the main way that ATP is made. And the second way is there is something called substrate level phosphorylation, which is really just a simple chemical reaction. And that happens in two places. Substrate level phosphorylation. And that occurs both in glycolysis and in Krebs. Okay, so more on that later. So the point of respiration, like I said before, is to take large chunks of energy, basically glucose, and break them down into small manageable amounts, ATP, that can then be used later. So let's start talking about how this occurs. Now at GCSE, it was just a simple one sort of line equation. What is it? It's glucose plus oxygen goes to carbon dioxide plus water plus energy. Unfortunately, it's a lot more complicated than that. It's no longer a single line. It's a lot of different processes. But let's try and break it down, first of all, with an overview. All right, so the respiration happens in a series of stages. Now, first of all, it's important to say that respiration can proceed in two different directions if there's oxygen or if there isn't oxygen. So first of all, if there is oxygen, respiration proceeds via an aerobic pathway, and if there isn't, it proceeds via an anaerobic pathway. In this video, first we're going to talk about aerobic respiration, and we'll come back to anaerobic a little bit later. Now, there are some similarities between aerobic and anaerobic. Both start with a process called glycolysis. Okay, so I want you to label this here, glycolysis. Now, if there is oxygen, then we proceed in this direction via the aerobic pathway. Let me just make that a bit smaller, actually. So if there is oxygen, we proceed this way via the aerobic pathway, and we go all the way all the way this way, kind of through this whole blue kind of highlighted line that I'm sort of highlighting here. And then we end up here. And in fact, let's put that in right now. So this is oxygen. This is oxygen right here. Okay, it seems crazy that, you know, oxygen, for the aerobic pathway, oxygen is only needed right at the very, very end. And oxygen is what's called the final electron acceptor, and it accepts electrons and hydrogen atoms to form water. So let's label that. Oxygen is called the final electron acceptor. But if there's not oxygen, then this whole process kind of backs up. It's almost like a traffic jam. Imagine if there's like a car crash here. If this was all like cars going down a little line, and there was some sort of crash or roadworks over here, all of this would back up with traffic. And at this, you know, maybe this is cars coming down and there's a junction here. Cars see that there's this traffic here, nothing's moving, so cars will go the other way. And it's a little bit like that. If you don't have oxygen, the final electron acceptor, then none of these processes work. So respiration would proceed in a different way. It would proceed via this anaerobic pathway. I'm going to try and highlight that in a different color, and that's this way. But no matter whether there's oxygen or not oxygen, glycolysis will always occur. So glycolysis is the first step of respiration, whatever the case. All right, what are the other steps? The other main parts are called the link reaction. Link reaction is that bit. Then we've got the Krebs cycle. Krebs cycle here. Perhaps you can be a bit neater than I am being. Actually, let me just zoom in even a bit more. I can get a bit tinier. And then finally, this stage here is called oxidative phosphorylation. I'll just leave it as oxidative phosp for short, but it's oxidative phosphorylation. Now, what occurs in each process? First of all, let's look at glycolysis. Okay, so glycolysis, it's in the cytosol. It's not in the mitochondria. It happens just in the cytoplasm or cytosol floating around in the cell. And glycolysis itself, we can divide it into 3 phases. So let's write those down. The first phase is called phosphorylation, where glucose is phosphorylated. The second phase is splitting, which is where the phosphorylated glucose is split into some 3 carbon molecules. And then the third phase is oxidation. And it's in this final phase that we do get a bit of energy released. In this final phase of oxidation, we do get 2 ATP made. And I'll put little brackets here, SL. That's substrate level phosphorylation. We also produce 2 reduced NAD molecules. Okay, hopefully you know what these mean, because if you're coming to this video as a revision tool, you should know a bit about this already, but we're going to go into that in more detail later. We'll get 2 reduced NAD molecules. Those can be fed into oxidative phosphorylation to produce more ATP later on. Okay, well, what's next? Link reaction. Okay, the link reaction is in the mitochondria. So it's in the matrix of mitochondria. And I'll kind of stretch that across here because that in matrix of mitochondria refers to both the link reaction and the Krebs cycle. Now in the link reaction, we just get, as a product, 1 reduced NAD. Okay, so I'll shorten that to 1 red NAD, red for reduced. Now the Krebs cycle, it's a little bit complicated, but per turn of the cycle, we get 1 ATP, we get 3 reduced NAD, so red NAD, and we get 1 reduced FAD as well, so 1 reduced FAD. Now, because that's per turn of the cycle, actually we get 2 turns of the cycle per molecule of glucose. So we can kind of put a bracket around that. We'll kind of times it by 2 almost, which would give us 2 ATPs, 6 NADs, and 2 FADs, which you could write down if you wanted to. All right, so what else can we add in here? I also better add in some waste products over here. I'm also going to add in the fact that we've got a CO2 kicked out here. We've got actually 2 CO2s kicked out here. I'll put a CO2 there and another CO2 there. And these hydrogen carriers, you can see there's 2H here. Let me highlight that in a different color, which I highlight it. I'll highlight it in green. So these 2H's here, those are these hydrogen carriers. These NADs and FADs are hydrogen carriers, and those are fed into the next stage. So I'll put this NAD and FAD, reduced NAD and FAD. Those are fed into the next stage where electrons from those hydrogen carriers pass down this electron transport chain, ETC. And what happens during that was basically we pump H+ ions into the intermembrane space of the mitochondria. And using that, we get this H+ gradient. So I'll kind of put a little box around this. And it's this H+ gradient that can be used to spin the ATP synthase molecule in something called chemiosmosis. Okay, so I'll put that again. It's a keyword, chemiosmosis. And I'll just do a little star there because that is all about this. Okay, this is chemiosmosis here. Down here. So we'll get to that later. We'll show you that process later. Okay, so that's the overview of respiration. Now, didn't talk about anaerobic respiration. Anaerobic respiration has glycolysis as its first step, but then there's another step, which is called fermentation. And that can proceed in 2 ways. So the top way is sort of in mammals or animals, and this bottom way is in fungi or plants. And we're going to come back to that later. I don't want to bombard you with too much information yet. We'll come back to that later. Right, so we're going to look at glycolysis now. But before we move on, I just remembered that I need to put a little brackets SL here. That's the ATP made in the Krebs cycle. That's substrate level phosphorylation, as well as in the cytosol. Okay, so let's look at that process of glycolysis. We'll pinch out a bit, we'll zoom out, and we'll move across over to here. Okay, so first of all, this molecule here is glucose. So glucose is the main respiratory substrate. So we'll talk about what a respiratory substrate is a little bit more later. But it basically means something that can be respired, something that can be broken down. Now we can respire other things, for example, amino acids and fats, and we do so in a very slightly different way. But the main thing that we focus on in A-level is looking at how glucose is respired. So it is the main respiratory substrate, and this one here is alpha glucose. Can we respire beta glucose? I suppose we can. Yes, we can. But digesting cellulose is difficult, but we can respire beta glucose as well. You just have to convert it. Okay, so I'm going to pop into a PowerPoint slide that I've got of this already. I'm going to be annotating around this diagram here. But already you might be able to see there's some color codes going on, and those color codes correspond to the 3 phases of glycolysis. The first phase is phosphorylation, the second phase is splitting, and then the third phase is oxidation. Okay, so the first thing we need to know is that this process, this breaking down of glucose, happens in the cytosol. This is nothing to do with mitochondria, so this is in the cytosol, and in fact it is a very ancient process, so it can even happen in simple cells which don't have mitochondria. So, what happens? First we start with glucose, which is a 6-carbon molecule. Then we add a phosphate onto that. So now this becomes glucose phosphate. And again, that's a 6-carbon molecule. So we've just stuck phosphate onto it. And then we add another phosphate onto it. So it becomes a hexose bisphosphate. So in some textbooks, you'll see that as fructose 1,6 bisphosphate, but you don't need to know that. It's just another hexose. So 6 carbon, 6 carbon, 6 carbon with 2 phosphates on. So how do we do this? Well, we have to add those phosphates from somewhere. So they're added from ATP. And actually, I've just realized I don't really like this here, because this is slightly incorrect. So the PI, I'm going to cross that out. It doesn't just float off. It's on the glucose. So we have ATP, and it gives up one phosphate to stick it on the glucose, leaving ADP behind. And then we have another ATP, it gives up one phosphate to stick it onto the glucose, leaving ADP behind. So we've added 2 phosphate groups. So this part of the whole process is called phosphorylation. So the purpose of that, really, just if you want to go into a bit more detail, is to kind of give the glucose, make the glucose molecule sort of almost a bit unstable, so that we can then break it down and oxidize it more easily. So we kind of put some energy in order to get more energy out. So there's a little note on that. So glucose is a stable molecule and must be activated by addition of phosphate before it can be broken down. Another thing to go even further is that when the glucose is phosphorylated at this first step here, it basically traps it inside the cell, so that glucose phosphate can no longer exit the cell via the same carrier proteins that it came in with once it's been changed into glucose phosphate. That's a little extra detail for you. Okay, so what does it look like? You don't need to draw this diagram. This is just for your kind of imagining of what it's like. That's what it looks like now on the left there. So it's a glucose. Well, actually, it's actually fructose now, but it has a phosphate on either side. It's a hexose with 2 phosphates on either side. So now we can split it. Okay, we can split it into something called triose phosphate, which is a 3-carbon molecule. You could almost imagine it pretty much as, just cutting it down the middle like that. It's not quite accurate, but it's pretty much like that. So we split the hexose bisphosphate into 2 times triose phosphate, and then we can get some energy back out of it. So we can get 2 things out of it. We can get ATP, and we can get reduced NAD. And in the end, we end up with something called pyruvate. So this last step is oxidation of triose phosphate to pyruvate. Now, if you're wondering about the numbers here, so we get 1 reduced NAD from each of the triose phosphates, and we get 2 ATPs from each of the triose phosphates, giving us overall 2 reduced NADs and 4 ATPs. Okay, so if you think about the overall ATP count from this process, we use up 2, but we get 4, so we have a net positive production of 2 ATPs overall. That's 4 minus 2. We also have those 2 reduced NADs that can be used later on in oxidative phosphorylation. So the enzymes that do this final process are called dehydrogenase enzymes, and that is something you need to know. A dehydrogenase enzyme is something that removes hydrogen. And then again, these ATP molecules there are made via substrate level phosphorylation as opposed to oxidative phosphorylation. Okay, let's take a quick detour into a little memory activity where we see if we can remember this. So normally when I teach this in class, this is what we do. Those are just the structures of triose phosphate and pyruvate in case you're curious, but you don't need to know those. So the memory activity is this. Can you explain this back to, well, your neighbour if you have a neighbour, or could you explain it back to me? Could you pause the video and basically say the sequence of events using that slightly blanked out schematic there? Pause the video, see if you can repeat back all the steps. Try not to look at your notes if you can. Did you get it? All right. Can we make it a bit harder? See if you can do it now. Using this schematic and removing that, could you do it now? Say it back to me if you can. All the steps, everything you know. Okay, sounds pretty good. You can repeat that as many times as you like and try and remember it. Now let's go back to our overall mind map and talk about the next stage of the process. Okay, so hopefully with those notes, your mind map looks a little bit like this. Okay, so all of that, remember, took place in the cytosol. So glycolysis occurs in the cytosol. So the bit of the screen that you can see now kind of sort of represents, I sort of tried to make it represent like a cell. So outside here we've got the cytosol, and then for the next part of the whole process we need to take this end product, pyruvate, we need to take it into the mitochondria. So I'll try and kind of draw a line to show that. Let's make a sort of purple dashed line. So this pyruvate is going to be taken into the mitochondria. Okay. And actually there is some energy cost involved in this. This is active transport. Into mitochondria. Into the matrix, in fact, of mitochondria. Okay, so we're inside the mitochondria now, where the next step of respiration happens. Now, this next step, remember, is only if oxygen is present. If oxygen isn't present, then something else happens to pyruvate. But if it is present, pyruvate gets taken into the mitochondria. I've got a PowerPoint slideshow which is going to go through kind of the steps of the link reaction and Krebs cycle. So let's jump across to that now. I'm going to be making notes inside this diagram of the mitochondria. Okay, so here we are in the matrix of the mitochondria, where both the link reaction and the Krebs cycle occur. Now, the link reaction starts with pyruvate. Remember, we just actively transported that into the mitochondria across the double membrane. And pyruvate is a 3-carbon molecule. Now, the pyruvate 3-carbon molecule is converted into a 2-carbon molecule, which is basically an acetyl group. And that acetyl group is stuck onto something called coenzyme A. So in that process, we lose a carbon because it's a 3-carbon becoming a 2-carbon. So CO2 is produced. And we also get the opportunity to reduce some more NAD. So now this diagram is a little bit simplified. Up here, you could think of, would have to have some oxidized NAD coming in and accepting the 2 hydrogens. And here, you'd have a coenzyme A would come in and stick to the 2 carbons from pyruvate to become acetyl coenzyme A. So this whole process, well, pyruvate we say is decarboxylated, that's a key word, and it's dehydrogenated. Now it's done by an enzyme called the pyruvate dehydrogenase complex, and it's a really big, like pretty awesome enzyme. Yeah, it's got like at least 3 enzymes, I think, working in like a little miniature production line. So it's kind of an enzyme does one bit, and then it passes it straight into the active site of the next enzyme, and then straight into the active site of the next enzyme. So it's quite an efficient process. So that's enzyme pyruvate dehydrogenase, which you need to know. Okay, so the acetyl-CoA, CoA doesn't go into the Krebs cycle, it's just the acetyl group. So CoA is coenzyme A. Remember, coenzymes, they bring chemical groups to enzymes to get them to do their job. So the CoA itself doesn't get involved in the reactions. It just is like it's like a shuttle. It's like a transport for the acetyl group. So what's next? Well, the acetyl group joins on to a molecule, which we'll come to later. It joins on to something which is already there, coming around this way, and it forms something called citrate. You need to know this one. Citrate is a 6-carbon molecule. So citrate, the 6-carbon molecule, it has quite a lot of energy in it, so we can extract some in the first step of the Krebs cycle. We can extract some reduced NAD. And we can also, we also lose a carbon at this point. We lose a carbon dioxide. So if you think, okay, with citrate was 6 carbon, if we're losing a carbon dioxide, the number of carbons must have gone down. And yes, you guessed it, it's gone down to 5. So we just call this a 5-carbon compound. Actually, we don't need to know the name of it at this stage in your studying. It's just a 5-carbon compound. So the 5-carbon compound, it loses a CO2. So we're going to go down to 4, aren't we? And again, it's got energy there. It's got hydrogens there, I should say. And those hydrogens can be kind of soaked up by NAD, forming reduced NAD. So now we have a 4-carbon compound because we lost the CO2. That 4-carbon compound, at this point, we can get some energy out of it by producing some ATP. And at this point, step 4, that is substrate level phosphorylation again. Now, as it's a slightly simplified diagram here, you might be thinking, where's that ATP coming from? Well, and ADP would directly take kind of the energy and the phosphate from that 4-carbon compound so it's a chemical reaction that's occurring there. All right, so we've still got a 4-carbon compound at that next step. Now, if you've noticed the color schemes here, you might be wondering, why has this green box got a little red line around it? Well, it's because it's a hydrogen carrier, but it's something slightly different. At this point, there's a different hydrogen carrier called FAD. So this is, again, another coenzyme. Okay, so NAD, coenzyme A, and FAD, in fact, ATP, these are all coenzymes. And reduced FAD, at this point, is the acceptor of the hydrogens. So now we have a 4-carbon compound again. Now that 4-carbon compound still has a little bit of hydrogen left, so we can take out some more hydrogens and produce another reduced NAD. And we've still got a 4-carbon compound up here. This one you need to know the name of, so that's oxaloacetate. So there's only 2 that you need to know the name of, actually. Oxaloacetate and citrate in that cycle, but then you just need to know that goes, start of the cycle citrate, then a 5-carbon compound, then a bunch of 4-carbon compounds all the way around to oxaloacetate at the top again. Okay? So we could do a little memory activity again. Once you've written that down, you can pause the video, and if I just obscure all those again, could you repeat all those back to me? Try maybe using your notes a little bit if you need to, then try without any of your notes, just using that blank template. Could you say that back to me? Okay, there we go. Now the thing about that you need to also remember is that per glucose molecule, the whole thing runs twice. Okay, so what have we made there? There so far we've made 1, 2, 3, 4 reduced NADs and 1 reduced FAD and 1 ATP. But per glucose molecule, because 1 glucose ends up being 2 pyruvates, we would make actually 1, 2, 3, 4, 8 reduced NADs, 2 reduced FADs, and 2 ATP molecules. And here's just another optional memory thing. You can skip this if you want, but if you want, could you put these boxes in the right order? Just pause the video, see if you can figure out which way around they go. And here's the answer. So first, the acetyl group released from acetyl-CoA combines with the 4C compound, and I'll let you look at those answers if you want to. And if not, we're just going to move straight on. Right, okay, so now we're going to tackle the final part of the respiration process, which is oxidative phosphorylation. Now, once again, this is only happening in the presence of oxygen, and this is where we use all those reduced NAD and reduced FAD molecules that we've produced. So just to sum up so far, if we look at this overall diagram up here, what have we made? Well, we haven't made much ATP. We've only made 2 ATPs in glycolysis, and then we've made 2 ATPs in the Krebs cycle, which is really not very much at all. It's kind of rubbish. So now is when we're really going to make lots of ATP. We do this in a bit of a weird way. We do this using something called the electron transport chain. Now, we're going to be labeling around this diagram here. So let's get into it. And once we've talked about that, I'm going to explain a little bit about how the structure of the mitochondria helps Krebs cycle, oxidative phosphorylation, and chemiosmosis all occur in synchrony. The first thing, I think, is to get some definitions down. And these definitions are pretty unwieldy, but I've done my best to kind of try and simplify them, because the definitions for oxidative phosphorylation on the internet or in textbooks are often a couple sentences long. So I've said that it is the formation of ATP by oxidizing reduced NAD and FAD in the presence of oxygen and the proteins of the electron transport chain. So for oxidative phosphorylation to occur, you've got to have some reduced NAD and FAD. We've already made those in glycolysis and the Krebs cycle. You've got to have oxygen. Remember, oxygen is what's called the final electron acceptor. And we've also got to have some specialized proteins that are in the inner mitochondrial membrane. That's this right here. And those are called the electron transport chain. So there's another definition which is kind of similar. It's called chemiosmosis. And this is more specifically just the final part of that oxidative phosphorylation, which is how ATP is actually made itself. and that's all to do with using a proton gradient. Sometimes it's called a proton motive force. It's just this kind of pushing power of a gradient of hydrogen ions that's built up, and it flows through this molecule called the ATP synthase molecule or enzyme. So let's look at this diagram now, okay? So the first thing is to kind of orientate ourselves. Where are we? So if you look here, the outer mitochondrial membrane is this membrane across here. It's a phospholipid bilayer membrane. And then we have this intermembrane space, and then we have an inner mitochondrial membrane here. Okay, so these are different compartments. This intermembrane space is its own unique compartment. The matrix down here is its own compartment. And then out here, this is kind of where the cytosol is, just above. So now let's look at the diagram down here, and let's look at this first green box down here, which you're going to label on your diagram. And this is reduced NAD. Okay, so reduced NAD could have been made in glycolysis, could have been made in the Krebs cycle, comes in, and it gives up the hydrogen atoms that it's been carrying. There's two of them. So reduced NAD can carry 2 hydrogen atoms. Now the thing about a hydrogen atom is we can split it. We can split it into its electrons and the proton itself. So that's what happens. The hydrogen ions are split one at a time into their protons and electrons, and they follow different paths. So now we can see what these different color-coded arrows mean, and a bit more about our sort of color code here, looking over on the right here. So these sort of circles, these yellow circles with a sort of orange outline, these are proteins. These are membrane proteins. They are kind of fixed in the membrane, so they're kind of hydrophobic. And they pass the electrons along. They're the electron transport chain. Red represents the flow of electrons down these proteins, and green shows movement of protons. Now, the thing about the proteins of the electron transport chain is they are arranged so that electrons move from a sort of more highly reducing protein down a sort of electrical potential gradient. So that the electrons, when they come out of NAD, they've kind of got high energy. If you studied photosynthesis, this will kind of sound familiar. They've got high energy and they move down a kind of redox potential scale. So they flow down, losing energy, and as they lose energy, that energy is used by the proteins to move protons and pump them. So as the electrons flow, protons are pumped into the intermembrane space. Now, there's 4 proteins of the electron transport chain. You don't need to know their names. Reduced NAD feeds in at the first protein. Reduced FAD feeds in the second protein. So kind of FAD is a little bit kind of not as powerful, if you like, in terms of generating ATP, because it just feeds into the electron transport chain slightly later. But again, the hydrogen ions on reduced FAD, it carries 2, just like reduced NAD. The hydrogen atoms, are split into the protons and the electrons, and then they flow down the chain. So what happens to the electrons when they reach the end of the chain? Well, they've got to be accepted by something, and they are accepted by that final electron acceptor, which is, of course, oxygen. So oxygen is an electronegative thing. It wants electrons, takes them in, and it combines with protons to make water. So we're looking down here. So the overall word equation for this would be 4E minus plus 4H plus O2 goes to 2H2O. Could write that in down here as well. Why not? So it would be 4E minus plus 4H plus O2 goes to 2H2O. Okay, so the final piece of the puzzle is ATP. How is ATP made? Well, we've actually made a huge gradient by doing this process. If you think about it, look what we've done. We have pumped 1, 2, 3, 4, 5, and 6 protons from the matrix, this is, remember, the matrix down here, into this space up here, which is the intermembrane space. So we pumped 6 protons kind of that way. And also, if we think about it, we've also used up these 4 protons that are kind of floating in the matrix down here. We've taken them and stuck them together into water. So overall, we've kind of created a gradient of 10 protons. We've removed 10 protons from the matrix, and we've added 6 over here. Okay, so that's quite a big gradient. Now that gradient represents energy. It's a kind of store of energy in the gradient, and that gradient can then be used to power this amazing enzyme. Now, as these protons here, these H+, flow down this channel, this is a little pore that can allow H+ to flow down, it spins and ATP is made. We're going to do a little bit more on that later, but the first thing I want to show you is a kind of animation of this whole process, okay? Because I think it's really important that you understand kind of conceptually how it's occurring and kind of see it for yourself. So let's go to that now. Okay, so here we are. This is a Harvard animation. It's really, really good. I'll share the link below so you can have a look at it on yourself. But I'm going to talk you through it. So these yellow, green, purple, blue proteins, these are the members of the electron transport chain. Now we can see that here we've got lots and lots of protons that are being pumped into the intermembrane space. Over here we've got fewer protons. The protons are the yellow sort of things that are pinging about. So we can see that there's a concentration gradient. Now we can try and focus on different members of the chain and look at the electrons moving along. So the electrons here are highlighted as blue, and we can see they come in and they flow down the chain, carried from protein to protein, before they finally end up at water, combining with oxygen to form water, and that would be sort of down here. Okay, so that's the whole process. This goes into a bit more detail. It just shows that actually within the proteins themselves, then there are a series of molecules that can kind of pass the electron from one to the other. So these are called redox centers, and they're all sort of arranged so that the electrons flow, kind of losing energy a little bit, piece by piece, and that energy is used to pump protons into the intermembrane space. Okay, so once again, I think I'll just zoom out back to the beginning just so you can see that whole process going. And if you want to look at this again, I really recommend it. I'll share the link underneath. So protons building up in the intermembrane space, and then over here we can see them flowing down this stalked particle, which is called ATP synthase, and making it spin. More on that later. Right, so hopefully your mind map looks a bit like this. We've now talked about oxidative phosphorylation over here. I just want to briefly touch on chemiosmosis, which is kind of the last part of oxidative phosphorylation, but specifically it's the flow of protons down the concentration gradient through the ATP synthase molecule, making ATP in the process. So here's a close-up of the ATP synthase molecule, which when they initially looked at the mitochondria with electron microscope, they saw these stalked particles, these little kind of bumps on the internal membrane of the mitochondria, on the inner membrane of the mitochondria. So they called these stalked particles. Later on, they realized that they were making ATP, so they renamed them ATP synthase. So the structure of the ATP synthase you don't need to know in detail, but let's just look at it a little bit. We have the sort of the head of the molecule. And you don't need to know this. This is post A level, but it's called the F1 part. You don't need to know that, but just in case you are curious. Then we've got this axial part here, which can turn. And we've got this F0 part, which is kind of the membrane part. So we'll just call it the membrane embedded. Hope you can see that. It says membrane embedded. Or F0 part, and then we have the head part, which is called the F1 part, and here we have a channel for H+. So H+ ions can flow down the channel through the membrane from the inner membrane space back into the matrix, and then they can kind of exit the channel this way. And as they flow, they cause the axle to rotate. So it's actually the axle that rotates inside, and as the axle rotates around, it causes the head region just to change its shape ever so slightly. And it's the head region where ATP is made. So what actually happens is ADP plus PI fly in and kind of stick to the head region. I'm just going to annotate that, right, sort of stick, and they kind of stick on the outside there. And they actually get sort of stuck there, and they won't move until that head kind of flexes as the axle rotates inside of it. So the head flexes, changes shape slightly, and that unsticks the ATP. So then ATP comes out. So let's have a look at that in motion to get you a better understanding. So over here we have an animation a few years old showing this process. I'll just take it back a little bit. So below we have the inner membrane space. We can see lots of protons. There's the yellow circles sort of flying about. We have that F0 region, which is kind of in the membrane. The grey molecules either side of phospholipids. That's a phospholipid bilayer. Now, the details of the proton movement you don't need to know for A-level, okay? So this is a little bit beyond A-level. But what happens is as the protons enter this channel here, they stick to the kind of barrel which is embedded in the membrane, and they make it rotate. And then when they exit, they kind of flow off this way. So the movement of protons into the F0 part and then out of the F0 part causes the axle to spin. Now we're looking down on top from the head here, and we can see that the axle internally, which is this blue region in the center, is kind of pivoting round and is slightly pushing on parts of the head. And as it pushes on parts of the head, you can see ATP molecules flying out. So we have sort of pink ATP molecules flying out, and once they've been sort of kicked out, you can see the ADP and PI molecules coming in, combining to form ATP where the ATP has just been left, okay? Here we have it annotated on the diagram. ADP and PI comes in, binds, converts into ATP, and then as that axle comes round again and dislodges or deforms the head shape again, then ATP will be kind of released out into the mitochondrial matrix. So this may look pretty slow in this view, but actually this is happening thousands and thousands of times a second. So we're really producing lots of ATP at high frequency. So let's go back to the mind map and just make a little side note on something that has come up in a few papers, which is called uncoupling. And this is kind of more of an application question. It's not directly on the syllabus, but it has come up a few times. So uncoupling. Anything that can allow H+ ions or protons to flow from the inner membrane, I'll write IM, inner membrane space to the matrix without going through ATP synthase will be a waste of energy, essentially. From intermembrane space to the matrix without going through ATP synthase. Now this wastes energy, and it wastes energy in the form of heat. So it means that instead of converting all that reduced NAD and reduced FAD into ATP, you basically convert reduced NAD and reduced FAD into heat energy. So what is this for? Or why does this happen? Is it useful? Well, it can be useful. So one way this can happen, there's a protein called UCP1, which I could add it onto this diagram here. You don't have to add it if you this is a bit kind of an application as a bonus thing. So if I added a UCP1 here, because this black protein drawing in there, if that could let H+ exit like that through the membrane without going through ATP synthase, that generates heat. So UPC1, sometimes called thermogenin. Maybe you recognize that word means making heat. So this protein is present in young babies, and in young babies, the sort of mitochondria in specialized tissue called brown fat tissue produces a lot of heat and less ATP. So it's kind of like almost like turning mitochondria into the radiators as opposed to ATP factories. So that can happen. That's one example of how uncoupling can happen. And there are also sort of poisons or toxins that can do this. One is called DNTP. or dinitrotriphenol. I admit that might not be the correct name, but it's definitely DNTP. And what it does is it's a chemical that, again, can move protons across the membrane. And if this actually was used as an explosive in World War II, I think it was, or maybe World War One, and people working in the factory, they reported that they were losing a lot of weight. They reported that they felt hot all the time, they were sweating all the time, and they became quite thin. So this was because a lot of their energy from their food stored in that reduced NAD and reduced FAD didn't end up going into ATP, and it ended up just going into heat. So they lost a lot of weight. They were really hot. Sometimes it's marketed as a diet aid, but it is extremely, extremely dangerous. There was an unfortunate case a few years ago of a young lady who died. I think it was in Bristol University from taking this DNTP. So that's just a little bit on coupling, which has come up in the past as an application style question. Okay, so we've looked at a lot of stuff about the mitochondria, about the processes that occur in the mitochondria, and also the processes that occur in the cytosol around the mitochondria, such as glycolysis. But now let's look at how the mitochondria, the structure of the mitochondria, is adapted to allow these processes to happen efficiently. Okay, so we're going to look at this diagram up here. Now that we know a lot about what happens in the mitochondria, we can relate the structure of the mitochondria to all these processes. So here we have a, this would be a this is a transmission EM, transmission electron microscope picture of a mitochondria, and here we have a diagram of the mitochondria. So what are we pointing at? Well, the first thing is the outer membrane. There is the outer membrane. And then I think just underneath this here is pointing to the inner membrane. I think. Yep. And there is a compartment between the two, okay, the intermembrane space, which we've already looked at. Inside we have the matrix, which is the kind of the fluid in the center of the mitochondria, which is where, of course, the Krebs cycle and the link reaction occur. And here we have these folds of the membrane called cristae. And I'm not quite sure what this is supposed to, I think this is supposed to be showing the stalked particles, actually. If we zoom in even more, it's supposed to be showing these little blobs on the inner membrane. So let's just call them stalked particles. And we can start to now kind of talk about how each thing relates. Okay, so first of all, the outer membrane and the inner membrane together are evidence of endosymbiotic theory, which is the idea, proven essentially. It's not really a theory, it's fact. The endosymbiotic theory is the idea that mitochondria used to be their own free-living bacteria, and then they were sort of swallowed by a larger proto-eukaryotic cell, so kind of early eukaryotic cell. And instead of taking it in a sort of phagosome or a phagocytotic vesicle and fully digesting it, it was kind of just kept in the phagosome. So the two membranes, the outer one kind of was the phagocytotic vesicle, if you like, and the inner one was the membrane of the original bacteria, but then digestion never occurred, and these bacteria just ended up living in our cells, providing us with lots of ATP. So endosymbiotic theory, we can link to that. The matrix, packed with enzymes. So the matrix, I'm also going to add some notes over here about other matrix as well. So the enzymes are responsible for Krebs cycle and link reaction, and also we produce large amounts of reduced NADH and of course FAD as well, reduced FAD. So we're producing these reduced hydrogen carrier molecules, and then we're using them up on that inner membrane again. So the inner membrane, I'll add some more detail over here. So it's the inner membrane here where proteins of the ETC, proteins of the electron transport chain, oxidize the NADH and the FADH. Okay, so it's made in the matrix and immediately broken down again or oxidized on the membrane. So those things can happen very easily. There's no great distance that the reduced NAD has to travel. So that's a benefit of the structure. The cristae create a large surface area. So there's a very large surface area for the proteins of the electron transport chain, and in fact for the stored particles. The fact that there's a folded membrane gives it a high surface area for all these things to occur. Now here we have DNA. Now this is going beyond A-level, but if you want, there are some genes on that loop of DNA. There's actually only about 13 genes on that mitochondrial DNA, which encodes 13 proteins. Most of them are the electron transport chain proteins. Now those proteins are actually quite large and complicated, but each one has at least one gene coded for on the mitochondrial DNA. Basically, the mitochondrial DNA can very quickly regulate the levels of all those different proteins to keep them in balance, because if any one protein becomes too high levels relative to the others, then it can generate something called reactive oxygen species, which causes damage to the mitochondria. So just having all those genes in the mitochondria makes for quick control. And then the stored particles, we know those are ATP synthase molecules. Just wanted to show you one quick diagram as well, which is here, which I think gives us another nice way of viewing this all. We can see that we've got these are the ETC proteins here. I'll just label them. You haven't got this diagram, but this is an ETC. Let's just call it 1, 2, 3 different members of the electron transport chain, pumping protons, which here are the red dots, into the intermembrane space. We get a pH gradient building up so that the intermembrane space is about the same pH as the cytosol, which is about 7.4. And really what we're doing is we're removing protons from the matrix, making it more alkaline, pH 7.9. That only seems not huge, it's only half pH unit. But remember, it is logarithmic, so that's at least, well, it's around about 5 times more protons in the intermembrane space than in the matrix. And then these ATP synthase molecules, often in mitochondria, they actually come in pairs towards the kind of end of the cristae that folds down into the matrix, and that's where ATP is made. Okay, so the mitochondrial structure and function are coupled. The structure supports the function of the mitochondria. Okay, so now we're going to circle back and talk about anaerobic respiration. Remember, most of what we've talked about so far has been about aerobic respiration, with the exception of glycolysis, which occurs both in aerobic and anaerobic respiration. So when there isn't oxygen present, pyruvate does not get taken into the mitochondria. It gets converted in either to lactate or into ethanol, depending on what type of organism anaerobic respiration is occurring. Now, what is the point of this? Why is this needed? Well, first, let's quickly focus on glycolysis once again. So let's look down here and look at glycolysis. What things are needed for glycolysis to happen? Well, we need some ATP to put in at the beginning. We need glucose. We need some more ATP here. And over here, we need some NAD. Okay, we need some oxidized NAD. Now, if glycolysis occurs, NAD is reduced. And if glycolysis kept on occurring, we produce more and more reduced NAD. But remember, without oxygen, there's no oxidizing the NAD later on in oxidative phosphorylation. It doesn't happen. So, in anaerobic respiration, we have to find a way of regenerating oxidized NAD. Okay? Otherwise, glycolysis wouldn't happen itself and we wouldn't get any ATP at all produced. So let's just add that up here. So fermentation, oops, that's quite large. I will just write extra step to oxidize, that's oxidized, to oxidized NAD. We could put brackets, otherwise glycolysis would stop too. Okay, so what are the two different ways? Well, lactate fermentation occurs in mammals. And ethanol fermentation occurs in yeast and plants. So let's look at the stages, or what happens in those two different fermentation reactions. Now in each reaction we're going to oxidize NAD, remember. So first of all, let's look at the mammal pathway. So this is quite simple. I'm just going to do word equation. You don't need to know the symbol equation. Pyruvate plus, I'll just write RID for reduced NAD. We convert it into lactate, which is lactic acid, lactate or lactic acid, plus oxidized NAD. Okay, so problem solved. We've regenerated oxidized NAD, and then we can do glycolysis. Well, what about in yeast and plants? Slightly more complicated steps. We have pyruvate, and the first thing is it is converted into ethanol. And I'll just draw kind of down here, we give off CO2 in that first step. And then we I'll kind of add going in there, reduced NAD. And then we use reduced NAD to hydrogenate ethanol to give it some hydrogen, which then turns it into ethanol plus oxidized NAD. Okay, so those are the two different anaerobic respiration pathways. Now, remember, in anaerobic respiration, we don't produce very much ATP. In fact, the only ATP that we produce in anaerobic respiration are these ones. Okay, that's the only ones there. I've just highlighted those in yellow. So in anaerobic respiration, we only produce 2 ATP, whereas in aerobic respiration, we produce around 34, I think. The figures sometimes vary depending on recent science. And here's just another figure to re-emphasize that. The only ATP that we make in anaerobic respiration is this bit up here. Whereas in aerobic respiration, the vast majority of ATP comes from the oxidation of these reduced hydrogen carriers in the electron transport chain and then chemiosmosis. So we're getting 34 ATP roughly from the chemiosmosis, plus 2 from the Krebs cycle, giving us a total of about 38. So aerobic, about 38 ATP, anaerobic, 2 ATP. So that's important to add onto our mind map. Let's do that. Let's do that now. So we could make a little note here. Anaerobic, no O2, low ATP yield. And maybe we could fit this in here. Aerobic with O2, high ATP. And that little graphic that I showed you, which I think is quite current, is 38. And this one is actually only 2. So aerobic respiration is clearly a lot sort of better if we can do it. Right, so one thing that can do both is yeast. So yeast can actually decide whether to do both, which one to do. So yeast is what's called a facultative anaerobe, which means it can respire aerobically or anaerobically depending on basically what's about. Or anaerobic. So one way to test kind of these different, to test the different energy yields of respiration, we talked about low yield versus high yield of ATP, is to let yeast grow in either aerobic or anaerobic conditions. So here's an experiment that you could do. I'm just going to try to draw a dotted line down there. I'm assuming I'll switch to black. So let's write experiment. and we would grow yeast in O2 and without, and then we'd give it some time, maybe a day or something, and then measure the cell density in culture, because this yeast would be growing like a little flask, a little bottle. So yeast, first of all, just to recap very quickly, if you look at this image I'm dropping in now, you can see yeast here growing through a process of budding. So that's just a little AS recap. It doesn't split via mitosis in exactly the same way as other eukaryotic cells do. A kind of smaller cell appears at the edge and buds off. Now, one way you can measure the concentration of yeast cells or other cells in solution is using a hemocytometer. So this picture down here is a hemocytometer. Let's label it. And I'm just going to drop in another image now of how that works. So it's a little repeating cycle of how it's working. So the first thing about when you position your hemocytometer slide, when you position the sort of cover slip on the hemocytometer slide, it's a specialized slide like this that has grooves and a space for fluid to collect in the slide. And then you position the cover slip over the top. And the cover slip has to be not too squashed down, not too sort of left up. It has to be at the right level. So there's an ingenious way that this can be calibrated. Basically, when it's at the right level, you get this rainbow pattern. It's caused by kind of diffraction of light, but it's called Newton's rings. So you get this kind of rainbow when you know the cover slip's in the right place. So rainbow / Newton's rings. Correct depth. Okay, and then you look at it down the microscope slide, and you basically count. You get this kind of grid that's like that a bit, and then this is kind of hatched. This is kind of hatched, and you count this square, this square, this square, this square, and the central square as well. You count those squares, and you basically count how many yeast cells are in it. So this could be an image here of yeast cells. You can see the video cycling on showing you how to do that. And you also have to make a decision that you generally you count cells. If a cell is lying on this line on the left or lying on this line above, you do count them. But if a cell is touching this line down here or this line down here, you don't count them. So you count the number of cells in that little grid. And then basically you can use some maths to work out the number of cells per cubic centimeter, okay? Because this is a set volume, so you use some maths to work out the number of cells per cubic centimeter, which gives you a sort of an estimate of how well the yeast has grown. So remember, this is all about an experiment, growing yeast. So at the end, you would see which solution has grown. Sorry, not which solution, which culture of yeast cells has grown the most, and you would expect that the culture with the most ATP available would grow the fastest. So you might expect the aerobically respiring yeast cells to grow more quickly than the anaerobically respiring yeast cells. So the yeast cells with oxygen would grow more quickly, giving you a higher count. Just a quick note, you might also, if you had two beakers, so this has just maybe cotton wool. I'm trying to draw this as cotton wool, so it's just kind of a loose boundary at the top to stop contaminates, but O2 can get in. This one you'd expect to be very turbid, okay? So turbid means cloudy because there's lots of yeast cells. Now you wouldn't be able to see the dots like this. This is way out of scale, but you might measure the turbidity. If you didn't have a cytometer, you could just measure how cloudy the solution was. And if you had a separate bottle growing with the same food source or whatever, but with like a bung in there to stop oxygen, so no O2, you'd expect that to be maybe less turbid, okay? Turbidity is another word for cloudiness, okay? So that's another way you could do this experiment if you didn't have a hemocytometer. Hemocytometer can also be used to measure the red blood cell count as well in a very similar way. That's why it's called a hemocytometer, because hemo means blood, cytometer means cell counter. So it's a blood cell counter, really. Okay, so that's one experiment with to do with respiration. We also need to look at another experiment with respiration, which is the respirometer. So that's this down here. Okay, so let's get in close with this respirometer and talk about how you would use it. Now, I'm actually going to sort of take away a bit of the diagram, and I don't want you to do what I'm doing just now. I'm just going to get a sort of fat white pen and kind of actually just rub out half of the respirometer. Is that white? Maybe that's white. So let's just pretend like half this respirometer wasn't here. I'm gonna get rid of this later on and talk about it. But let's just pretend that we only had a very simple respirometer, which was just one tube connected to some living things. Okay, so we can actually label what I'm about to do now. So here we have a living thing to be studied. And let's say in this instance that it's wood lice. Okay, it doesn't have to be wood lice. It could be germinating cress seeds, it could be anything. Oops, I put wood lice. Okay, so wood lice. Wood lice respire. Wood lice respire and they take in oxygen and they give out carbon dioxide. So you might think that if they're respiring, they're taking in, I don't know, carbon dioxide and oxygen at the same rate, then there would be no change in this, in the volume of this, of the air in this tube. Well, we can kind of tweak the system so that we can measure changes in gases. And what we do is we put here, again, you can label these things that I'm labeling, sodium hydroxide solution. Sodium hydroxide solution. Now what this does is it absorbs CO2. So now any CO2 that is produced by these wood lice that are probably respiring aerobically is absorbed, which means that oxygen is being taken in, and any carbon dioxide that's been produced to kind of replace it is also being taken in by the sodium hydroxide, which means that the pressure in here decreases. And by the way, these are kind of sitting on a little platform. Okay, there's a little platform here. It is a gauze, G-U, how do you spell gauze? GAU, sorry, GAU, gauze platform, which is like a little wire platform to allow gases to move through into the sodium hydroxide. You don't want the wood lice in the sodium hydroxide because it's an alkali and that's unethical. Okay, so there's ethical considerations how to kind of treat living things. So the pressure decreases. So I'm just going to write P for pressure, decreases and goes down. Now that would draw fluid into the tube. Now don't label what I'm about to do. Don't label what I'm about to do. But the fluid would be drawn up this way. Okay, because air would be kind of rushing in down this way. Air would be coming in the end of the tube and the fluid would be moving up. Now that's great, but there's a big problem. If you want to do experiments like changing the temperature of the wood lice and seeing how that affects their respiration, then you've got a big problem. Because if you took that whole tube and you imagine you just dunked it in a beaker of warm water, maybe water at 40 degrees Celsius. It's a bit warm for the wood lice, but I think that would be ethical. Yeah, it would be. Would it? Okay, let's not do 40. So let's say we try them at 10 degrees Celsius, 20 degrees Celsius, and 30 degrees Celsius. In their normal range of temperatures they would experience, that's ethically kind of sound. Well, the problem is that if you put that this air in the tube in the different temperatures, the air would expand or contract. It would contract a bit in cold temperatures, it would expand a bit in warm temperatures. So that would be a factor that would also affect the movement of this coloured fluid in this respirometer tube. So therefore, what do? Well, we double the setup. Okay, now I'm going to take my eraser here and rub out all that stuff that I just did. Okay, might rub out that as well, yeah. Right, so if we double the setup, this is a double respirometer. Now what we have is we have another respirometer basically connected via this U tube. This is a U-shaped capillary tube with a fluid in the middle. So let's label that now. So this here, U-shaped tube, and it contains manometer fluid. I'll put that in red because it's red there, which is basically water with some red food coloring in it. And it just means you can kind of easily track the movement of it. So what you would expect in this setup is if you you connect both sides. Now both sides are at the same temperature. Now, and this side, to make it a very good control, you want to have the volume of the air in this tube needs to be exactly the same as the volume of air in this tube. Now, when you put wood lice in there, you actually decrease the volume that's remaining in the tube for air. So whenever you put living things in your test tube, like on the left, you want to put something that has the same size and volume as them on the right. And use glass beads. Okay, so these things here are just glass beads with about the same volume or mass as your test subject. Glass beads with, now in your textbook it says, well, my textbook it says same mass, but I think same volume is more accurate. Glass beads are same volume. And you have exactly the same stuff. You have NaOH, that's sodium hydroxide, I'm just using chemical shorthand there, solution there. You have exactly the same setup. Now, these little openings up here are a little bit complicated, but basically when you're doing the experiment, you need to kind of close off the tube so that air can't be pulled in this way. And actually this one you leave open so that air can be pulled in here. Because what you expect is as the pressure drops here, you would expect the sort of the higher pressure over here to push this fluid round, so the fluid would move up, and you can add this on your diagram, so the fluid kind of moves up like that, and this fluid down here would go down. So let's change pen color, and I can kind of so that that's gone down a bit, and you would measure that change over time. Now here's a little great video which shows a time lapse of this experiment. So here's an experiment happening, and you can just about see in the background there as that kind of cycles on. In this experiment, there's maggots in the kind of live organism tube, and there's glass beads in the other tube. You can't see anything moving in the other tube. And you can see that over time, the fluid moves around, showing the rate of oxygen use. Okay, so that would be a rate of oxygen use per minute. So I'll just maybe kind of annotate here. Use scale. Whoops. Scale, not sail. Use scale to get rate of O2 use maybe per minute or even per second if you wanted. Sorry, that's a bit messy. Rate of O2 use per minute. Now, often you also do this per unit mass of the organism, so you might do rate of oxygen use per minute per gram of wood lice potential. Now, the great thing about this double setup is that if you wanted to, again, see how temperature affects the wood lice, you put the whole setup with the two tubes in some 10 degrees C water, in some 20 degrees C water, and some 30 degrees C water. And because each tube is expanding and contracting the same amount, because it's got air in it, expanding and contracting due to the temperature, then it eliminates the effect of this expanding and contracting gases on the movement of the U-tube. So it's a control. It's a way of controlling things to get a better result. Now, the very last piece of the puzzle is how we can use this respirometer to actually work out something called the RQ. So this is the respiratory quotient. And what we can do using the respirometer's quotient is we can first work out the amount of oxygen that is absorbed, and then we can just remove the sodium hydroxide. So then we can run the experiment again and work out the amount of carbon dioxide produced. There's a bit of maths involved, but you can do both things, okay? So the RQ value can then be calculated. And the RQ value is defined as the volume of CO2 given out, so I'll just write CO2 out, divided by the volume of O2 taken in or absorbed. Okay, so that is the RQ value. Now, by recording those numbers and calculating it, we can work out actually what is happening in those wood lice. What are the wood lice respiring right now? What do they have for breakfast? If the wood lice are respiring aerobically, then they would have an RQ value of a maximum of 1 or below 1. And if they're respiring carbohydrate, they'd have an RQ value of 1. If they were respiring protein, they'd have an RQ value of about 0.9. So I'll put this little wiggle there because it's around 0.9, not exactly, but around. And lipid, again, around, not exactly, but 0.7. Why is that? It's all to do with the stoichiometry of chemical reactions involving respiration. You may remember that respiring glucose is 6O2 plus glucose goes to 6 CO2 plus 6 H2O. So if 6 O2 is taken in and 6 CO2 is given out, we'd expect it, have a 1-to-1 ratio of CO2 out versus oxygen in, and therefore we get 1. But if we use different chemicals, the ratio, the chemical reaction, the stoichiometry of it changes a bit, which gives us a different number in the ratio of carbon dioxide produced over oxygen in. So, and if oxygen isn't being used, if there's anaerobic respiration going on, then the RQ value would be bigger than 1. So above, that's the mathematical symbol, above 1. So we can tell a lot using the RQ value. Right, last thing we want to link to is kind of links to RQ value is, well, the fact that we can respire different things. We don't just have to respire carbohydrate, we can respire different things. So we're just going to fill out this table. So carbohydrates, they're polysaccharides, and they're polymers of essentially something like glucose. Okay, so that's glucose there. Or we can have polymers where we have, branched polymers. But how is it respired? Well, first digested into monosaccharides. And there might be isomerization. So you can respire stuff like fructose. But fructose, which is a hexose sugar, can be converted into glucose, and then we respire it in the normal way. Or something like even you could break down something like ribose or deoxyribose as a pentose sugars, but you could isomerize them through complicated chemical kind of maneuvers, and then you could respire them in the same way. So the energy value of carbohydrates is about 15.8 kilojoules per gram. I'll write in the other ones as well. For lipids, it's 39.4, way higher. And for protein, it's around about 17. Again, it's kilojoules per gram. Kilojoules per gram. Now, why is that? Well, we have to think about the structure of the lipids. I'm just going to do an outline structure here. A lipid is glycerol, and then you've got the fatty acids, okay, attached to them. I'm going to do this the way around because I don't have space. Okay, so there's a long chain. The fatty acids are have lots of CH bonds and lots of hydrogen atoms and little oxygen. Now that makes them a very good store of energy, because if they have little oxygen, it means there's not wasted space. In a sense, if the molecule's got oxygen in it, it's a waste of space, because you can get oxygen from the air to react, to kind of do the respiration. You don't need oxygen in the molecule. So lipids are very energy dense, and they are respired by beta oxidation. I don't know why I changed colour there. I do that sometimes. Beta oxidation, what it does is it chops Fa, which is fatty acid, into 2 carbon chunks, and then they go into acetyl-CoA. Okay, so I'll just zoom out and show you where that occurs. So we could kind of put it into here. So we could also have, we could add it in if you want to. You could put fats, beta oxidation, and then we feed 2 carbon chunks into acetyl-CoA, and then they go around Krebs cycle and be respired that way, giving a very high energy yield. And then for proteins, remember that they are polymers of amino acids. So let's just draw an amino acid. Now an amino acid has its carboxylic acid bit, has hydrogen here, and it also has its amine bit. But the most key thing about these is they have this R group. So this R group varies, and that's why you can never say that the RQ is exactly 0.9. It varies depending on what amino acids are in the protein, basically. And proteins, generally, the amino acid itself, so how is it respired? So first of all, digested again into amino acids, alpha for short. And then the amino acid is first deaminated. You should have covered this in the excretion unit. They're first deaminated, and that puts them into something called a keto acid. And the keto acid, or Ka, I'll just write down here, can go into Krebs, pretty much, okay? Normally it goes into Krebs cycle, but it goes into different places depending on the R group. So that's again why it has a varying RQ value, and I think maybe a bit of a varying energy yield as well, depending on where it goes. So, for example, around here, you don't need to know this much at all, but I think it is probably in your textbook, that you might have some amino acids might feed in at this point. Some amino acids might feed in at this point, depending on the size of the R group. Some amino acids might feed in at this point, which means that we have a kind of a varying energy yield and varying RQ value. So we've done it. We've reached the end of respiration, which is quite a big, complicated topic. There it all is. Very briefly, review. ATP, universal energy currency molecule, small pieces of energy. Overview of respiration. Glycolysis can happen with aerobic or anaerobic. Then link reaction Krebs cycle oxidative can only happen with oxygen. Fermentation is an extra step to regenerate NAD if you don't have oxygen. Glycolysis takes glucose and turns it into pyruvate. You get a bit of ATP, but much more ATP is produced in the mitochondria. Link reaction and Krebs cycle generate reduced hydrogen carriers, and then they are oxidized through oxidative phosphorylation and chemiosmosis. The structure of the mitochondria tells us that it used to be its own free-swimming bacteria. We call this endosymbiotic theory. And one main thing about the mitochondria, got a lot of membranes, a lot of surface area there for the reactions of respiration. Experiments to do with respiration, we can do experiments with yeast. We can use a Hemocytometer to measure how yeast grows with oxygen or without oxygen. We can do experiments with bigger animals like woodlice or germinating seeds like cress seedlings. We can use a respirometer, and we can either use a single kind of respirometer, or we can sort of use a double respirometer, which controls for changes in the gases in there due to temperature, for example. We can't just respire glucose. We can actually respire a range of substrates, such as carbohydrates, lipids, and proteins, by breaking them down biochemically into monomers, first of all, and then feeding them into this whole respiration pathway at different points. Lipids have the most energy yield per gram because they've got a lot of carbon and hydrogen with little oxygen. That's it. I hope you've enjoyed the video, and I'll see you later on. Bye-bye.